This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on September 25th, 2019. I'm Vincent and you're listening to the podcast all about viruses. Today we are recording in Reno, Nevada. We are at the University of Nevada, and I have with me 4 faculty members. Are we all at the medical school? Our appointments? Yes? No?
Biochemistry.
Biochemistry. So there's a mix of medical school and non-medical school appointments. So let me introduce them to you. On my left, the person who invited me to come here, Cyprian Rossetto.
Welcome. Hi, welcome to Reno.
With one of the best names I've heard.
Thank you. Thank you.
You like my name, Raccaniello?
Absolutely.
Okay, we'll find out a little bit about your background. Next to her, Dave Aucoin. Welcome to TWiV.
Thanks for having me. This is great.
Pleasure. Over on the other side of the table, Subash Varma. Welcome.
Thank you for having me.
My pleasure. And finally here, so all of the 3 so far have been in micro and immunology, right?
Correct.
And now we have molecular biosciences, Claudia Rooker. Yeah. Welcome. Rooker, that's really a famous name in virology.
I know.
Do you know Roland Rooker?
I just listened to the TWiV you did with him.
Oh, good.
Yeah.
He's awesome, isn't he?
Yeah.
But you have a U with 2 dots.
Yeah, he just changed his 2 dots into the UE, which is what I do for paperwork as well, actually.
So it's pronounced the same, Rooker.
Rooker, yeah.
Nice. Well, welcome to TWiV. And we're going to talk a little bit about your work and what it's like being here in a town with lots of casinos. So when I flew in yesterday, I go to the airport, you walk through and they're stuff, people are gambling immediately.
Slot machines.
Slot machines and other stuff. And people are sitting there using them. And then, of course,
coming and going.
Downtown you have all the big casinos. However, Reno has a university, which is pretty cool. And I guess so does Las Vegas as well, right?
Yes.
Where the other campus of the university is. And there are lots of neighborhoods in this town that seem to be really nice. And I had great breakfast there this morning with some students. So there is normal life here outside the casinos.
Yeah.
Probably you never go to the casinos.
I haven't seen them. I don't know since I moved here.
Yeah.
You probably should stay away.
Yeah.
It's a small area downtown.
Yeah.
But Reno's a very old city compared to Las Vegas, for example. There's we have houses here built in turn of the 19th century. So nice old neighborhoods, nice new neighborhoods. Outdoors, the lake, hiking, all sorts of stuff.
Yeah, and also we like to call this as a college town as compared to Vegas. That's a casino town. We are more like a college town kind of thing because university is bigger than, I think, in terms of number. We have a lot of students here, undergraduates, and the med school is good here, so it's a great town.
Yeah, one of the students mentioned that you consider it a college town.
Yeah.
He didn't agree. He said Berkeley is a college town. But you can call it a college town. That's fine. So I guess you have your little corner of town where it's academic, right? That's the university.
Yeah.
Right. Can you tell that there are a lot of students around? Because in many college towns, in the summer, it gets deserted.
Yes, we can tell because in the summertime, most of the places they get empty, and in the time when semester is on, it's hard to find any apartment around the campus. All are taken, so that's one criteria. And the traffic also is a lot lower in the summertime, but spring and fall when semester is on, traffic is kind of low. It gets crazy around the university. Yeah, so that way you can tell it's affected by the students who are here in the fall and springtime.
In New York, we have many universities, right? Oh, probably over a dozen, and you cannot tell. Students are not. You can't tell.
Yeah.
It looks just as crowded and everything, but in a smaller town, you can tell. I'd like to start by finding out a little bit of your histories, where you're from and where you were educated and so forth, what brought you here. start with you, Cyprian. Where are you from?
So I'm originally from Redding, California. It's Northern California. And so when I say Northern California, I mean way north. So not the Bay Area. It's
North of the wine country?
Yes. Yeah, north of Sacramento.
It's affordable country, right?
Yeah, yeah. It's in the foothills. We're about an hour and a half before the Oregon border. So,
oh, yeah. I drove once from Crater Lake all the way down, so I went through there. I have a colleague at work who says that's the only affordable place in California. You have to go quite far north, though, right?
Yeah.
And it's beautiful, right?
Yeah, it's beautiful. In the summer, it's quite hot, so, over 100 and 110. But yeah, it was great. So I was born there, I was raised there, went to high school. Went to high school there, graduated from Shasta High. Actually, so then as far as like my career in science, in between my junior and senior year of high school, I was part of the NSF Young Scholars Program. And so I actually got to go down to UC Davis. I spent 10 weeks at UC Davis, lived in the dorms as a high school student, and worked in a lab for the summer. And that was just so eye-opening. For me, I always loved science, but, I just didn't really understand what it was to do research. And so to be able to go into a lab and work there, during the week, on the weekends, and to really get a sense of what it meant to be a scientist, that was kind of what really hooked me. I mean, I had some great science teachers in elementary school and high school, but that was really, it hooked me. And also to be on a campus, like in that academic environment, was just amazing.
So you say you always liked science. Was there did either of your parents do science?
No, neither one of my parents are scientists. I do remember very specifically in 7th, 8th grade, we had a new science teacher that came to our school, and she had been trained as a chemist. And this was her first years being a teacher, and so she brought in kind of all that chemistry that she had been doing. And I remember she had a friend that gave her just LB auger plates. Everybody in the classroom got a plate, and we got to choose whatever we wanted to go streak out. And again, that was something that was just amazing, because you don't see these microorganisms. You streak a plate, and then they're there. So after that, I was pretty much hooked. The other thing that was funny, too, that happened during high school, and I found this again when I was going through my bedroom back at my parents' house. So the Ebola outbreak in 94, 95, I was in high school, and I actually wrote a letter to the CDC, and I asked them for information. And so I found the packet of information that they actually sent me.
They responded.
Wow.
Yeah.
Great.
All right.
So, it was kind of they sent me a little form letter and
Sure.
thanked me for my interest. And then they had sent me photocopies. I think it was probably Fields Virology or something, but they sent me information about Ebola. And that was the other thing in high school. I was so fascinated by the Ebola outbreak. I read The Coming Plague and Outbreak. I went to see the movie Outbreak when it first came out, and obviously that's not, that's not like real science, but I still just loved that. And,
so you decided to go into science after that experience, right?
Yeah.
So then where'd you go to college?
So I did undergrad at UC San Diego, and again, it was one of those things where I wasn't necessarily sure where I wanted to go to school. I wanted to go somewhere that had like a strong life science departments and things like that, strong research. And it was one of those things where I went to visit the campus, and you go there and you see, Salk and Scripps and all the research that's going on down there. And that was it. I was sold. And so I went.
Bio major?
Yes, I was a biology major. As much as I love microbiology and viruses, I really loved all biology. And so I stayed as a general bio major so that way I could take any biology class that I thought was interesting, from ecology. And it was great because down there, you could take ecology courses, and we would go down to the tide pools and, look at the stuff in the tide pools and the starfish and things like that. And so while I was there, I got involved in undergraduate research. So I volunteered in an immunology lab, and it was fantastic. They were wonderful, patient, and I just loved doing research.
So you decided then you would get a PhD?
Not right away. So I had done 4 years, and I was at the point where I needed to apply for graduation, and I didn't want to graduate. I just love school. So then I had to convince my parents that I wanted to do a second major. So even though I was complete with my bio major, I decided to stay for an extra year. I did a second major actually in history. And at that time, I took a lot of history of science courses. So then I did finally graduate after 5 years with bio and history. And I stayed on as a technician in the lab.
Same lab you had been working in?
Same lab, yeah. It was an immunology lab. I just loved what I was doing, and I really didn't know what I wanted my career to be. And so they said, why don't you stay here? You're doing good work. And so I stayed there. And
did you get published in that lab? Yeah.
Yeah.
Who was the PI?
So that was A.L. Roz. When we worked on immune modulation using different TLR ligands, mainly CpG DNA. So I was there as a technician, and then I decided at a point, I love what I'm doing, but I want to do more and, really kind of determine what things I get to research. And so I went to grad school.
Where?
So actually I went to grad school here. So again, it was one of those things where I applied to a few different places, was interviewing at a few different places, and came here. And it was really just talking to the faculty. They were so excited about their research, and just wanted to know about me and what I was interested in. And it just felt like a really good place. And so I came here, and
I guess you wanted to stay local, right?
Yeah, I do like the West Coast.
Because most of the students I met last night for dinner and breakfast are pretty much all from either Nevada or California, right? People stay. So you take advantage of that here at the university, people who want to stay, right? Who'd you work with?
So I worked with Greg Perry. So I came here, I was in his lab, and so then after I graduated...
What was that? What did you work on?
So for my PhD, I mainly worked on Kaposi sarcoma herpes virus, so KSHV. And I looked at DNA replication, both during a latent and lytic infection, and kind of the balance between those two states of the virus. I also did my postdoc with him as well, but at that point I transitioned to working with human cytomegalovirus and looking to see what factors are involved in latency.
Okay. And then you became a faculty member after that. How long ago was that now?
So it's been about 3 years.
Okay.
Yeah.
So you're pretty new as a faculty member. Okay. So we'll talk about your work. Now let's hear from Dave. Where are you from?
So I'm from Massachusetts, Braintree, which is a little town south of Boston.
Yep.
Grew up, got interested in science, I think just kind of same way, like reading things like The Hot Zone and cool movies and things like that, and hearing about the CDC and where they were running off to, solving mysteries. And So undergrad, I went to UMass Amherst, was interested in science. I was actually an exercise science major, so exercise physiology, and really enjoyed my first kind of physiology course, and that happened to be an exercise physiology course, so I thought that's probably what I wanted to do. So went through UMass Amherst, became a kind of a more, I would think more like an exercise it was an exercise physiologist position, but it was more like working in a health center, like helping people work out and making sure they were, using the equipment properly and things like that. Didn't really enjoy that as much as I wanted to, so actually got a job at Children's Hospital in Boston and worked in a pulmonary lab. So we were measuring the lung function of patients that had asthma and cystic fibrosis. And I think what probably got me hooked the most was working with cystic fibrosis patients and learning about how they're, chronically infected with different types of bacteria, Pseudomonas, Burkholderia, for example, and probably I think one of the saddest diseases where patients are, in their prime are dying of this genetic disorder. So. Worked there for about 3 years, was really interested in the science behind what was going on. It's a genetic disorder, cystic fibrosis, so I actually applied for some graduate programs, one being at UNR, actually. My wife is from Reno. I met her on the East Coast. And so I started off in a lab that studied chromosomes and just chromosome segregation. Did all sorts of studies there for a couple of years, but then decided to apply to the molecular biology program or the cell and molecular biology program, and wound up in the same lab that Cyprian was in as well. So I worked with Greg Perry, studied Kaposi sarcoma-associated herpes viruses, kind of where on the DNA the virus initiates its lytic replication from, so kind of map that out along with doing a few other things. So I found that really interesting. Love working with viruses. Went ahead and graduated and went off to Stanford, did a postdoc in their microbiology and immunology department under Ed McCarskey. Yeah, and great guy. And studied human cytomegalovirus there for a while. How does the virus kind of escape from the cell? And looked at a few viral mutants, trying to figure out, what was the function of these viral gene products. And then actually, at that point, we were living in Palo Alto, had 3 kids. Life got really expensive. Rent got really expensive. So was kind of looking for different positions and wound up back in Reno. Did a couple of years of postdoc with a fellow named Tom Kozel. This was immunology, kind of diagnostic discoveries, things like that. Really enjoyed doing that. And so that's what I currently do now. I'm a full professor. I'm the chair of the department working on antibody-based diagnostics and therapeutics.
So I looked at a job at Stanford way before McCarskey went.
Okay.
I decided to go to Columbia. I couldn't handle going to California because I'm an East Coaster,
Sure.
But I would have been there if I had taken the position. You would have known me a long time ago, probably.
Yeah, interestingly, same situation. I was looking at postdocs, and I loved this one lab at Harvard, Dave Knipes' lab.
Yep.
And had the opportunity to potentially go there. It just came down to where did my wife actually find a job? She was a school administrator at the time, and there was a position open at Stanford. It was like 50-50. I really loved Ed's lab, really liked Dave Knipes' lab. Almost wanted to get back to the East Coast, but I think I'm glad I stayed in the West Coast. I love Reno. I have my list of reasons why I'm here, and it's no mosquitoes, no humidity, not a lot of ticks for one thing, and not a lot of traffic, and we don't shovel a lot of snow. So
that's good. It's all good.
It's
the traffic
all,
part I like.
Yeah,
I'm dying from traffic.
Every time I travel back east, I wind up in a traffic jam, and I realize how nice it is here. So, but I love the East, but.
We'll have to talk about the no mosquito thing.
There may be some coming our way.
So last night, one student said no mosquitoes, but this morning someone said there are some. But I was going to ask you about that.
Right.
Yes. So Subash, where are you from?
So I'm from India.
Which part? North?
The north part of India, near New Delhi.
Okay.
So I did my PhD, like undergraduate and graduate work in India. And I came here for a postdoc at University of Pennsylvania with Dr. Earl Robertson. He works with the herpes viruses like Epstein-Barr virus and Kaposi sarcoma virus. But before getting to that, I want to give a little more background on my in India time. So I did my undergraduate, like a biology major, and then from there on, there was a new program, like in master's in microbiology, and that was highly competitive. And there was an entrance exam. You got to qualify to get into the program. Only like top, whatever. Like there were only 20 seats for the entire program in the university, so I made it in there. So I was not going to do any like masters or something, but since the program was so competitive, people told me that you got to do this thing. This is a good program. So I ended up in microbiology just because I qualified in there. And then I did some work there on like fermentation, and I was fascinated with the fermentation, like how yeast though they can ferment like sugar to alcohol and all that, even though I didn't drink at the time. It was fascinating, like how those things happens. And then from there on, we heard about anthrax, HIV, and all those kind of things. Like it seems very fascinating to me, like how viruses can deplete the immune cells and all those. We didn't do a lot of experiment there, but like it's just fascinating, like how a virus can kill the cells or deplete the immunity or the immune cells and all. So then I decided to do PhD from there on, because master's wasn't enough to get anywhere what I wanted to do. So I ended up in a lab where they were studying bacteria, like how bacteria enters the cells, like what kind of mechanism it utilizes to get inside the cells. And that work was pretty interesting. One of my highly cited papers is still from my PhD, like I don't know how much citation it's got. So it was pretty good work. And from there on, I also went to Italy, University of Florence, to do some collaborative work. I was there for 3 months, and there we used the technique which we did not have in India. So when I came back, I submitted my thesis and got awarded the PhD, and we published, I think, 5-6 papers out of my PhD. Then it was time to look for postdocs. So that's when I contacted Earl, and he was working with viruses. So since I knew that certain things go in the cells and they get kicked out, but viruses, like especially herpes virus, they go in the cells and they attach inside the cells. So that was fascinating to me, like how these viruses can attach inside the host cells. And then he asked me to write some synopsis, like, what do you want to do in my lab? I said, okay, since I was interested in the part, like how the virus attaches, I looked at ABNA1, that's a protein which can help as an anchor protein to retain the extra chromosome in the cells. So I told that this could be used as a therapeutic kind of a approach where you can put some genes and they can probably be there. So he was impressed, and he said, okay, come over. And then I came to his lab to do postdoc. And there he wasn't funded too much on EBV at the time, but he asked me to do work on Kaposi sarcoma, because there was similar virus, and in this case, we worked with another homologue of the protein called LANA, and the LANA protein attaches KSHV, the Kaposi sarcoma virus, to the host chromosome. So we did a lot of work figuring out, like, what are the molecular mechanics, how the protein helps in attaching the viral genome to the host chromosome. So it was interesting. We were highly published there. We did also identify what sequence is there to initiate replication. And at that time, when I was postdoc, I got promoted to research associate. And then I was awarded this K99 grant, which is a transition award. So that helped me to find job. And I was offered job at a couple places, but I ended up coming here to Reno because Greg Perry, who was the former chair here, he convinced me that this is a good place, and I had never been in the West Coast, but he said that this is a good place, and I'm happy that I made the decision to come here. Reno is a nice place, and have been here since last 10 years, and 10 years. It's pretty...
Did he use the mosquitoes as one of the attractions?
Lack thereof.
I wasn't too. I mean, I wasn't thinking of too much about mosquitoes, but yeah, it's a good thing not to have mosquitoes. That's definitely.
No, it's clearly the people in the science that attract you, right? Yeah. So, but I noticed you published together, right? Cyprian Subash on KSHV, right?
Yeah.
By the way, KSHV discovered by Pat Moore and Yuan Chang at Columbia.
Yeah, actually 25 years ago.
Really?
Yeah.
We were very sad when they left.
Yeah, they are at UPitt now.
Pitt, yeah.
Yeah, UPitt, yeah.
Yeah, Pat's been on TWiV a couple of times.
Okay.
Yuan doesn't want to come on; she's too shy. But Pat comes on, and he likes to talk. He's very fun. So Claudia, where are you from?
I'm German, born and raised, and I don't.
Which part?
Sort of southwest. So the village I grew up in, for most of my life at least, has 130 people. So you're not going to know that.
What does it say, near Munich?
It's about an hour and a half from Frankfurt. So it's kind of
more north.
There's Würzburg, if you've heard of it.
Yeah, I've been there actually.
Yeah, okay, that's the closest.
It's a good university in Würzburg.
Exactly, and that's the closest kind of city that we have. Yeah, it's like 45 minutes from there.
Okay.
I can't really fully identify with the story of, I always loved science and, this was what I wanted to do from, age 5. I think it was more, I liked animals and I liked being outside and I probably liked science, but I didn't think about it. I don't think I had a concept of the fact that there are scientists until I, started university. So I just ended up in, grade at the time where you apply for university or not. And I knew I wanted to go to university just because I liked sort of studying. And my brother, for example, didn't decide to. He didn't want to, sit in rooms and study all day. But I did. And then I was at the point where I'm like, what do I like? I like biology and I like English. Well, what can I do with that? So my options were, in Germany, you can do a teaching degree and then you choose 2 subjects. So I kind of considered that for a moment to do biology and English as a high school teacher, essentially. But then I don't think I couldn't imagine just being sort of stuck in that life with huge classes of kids every day. I feel like a lot of the older teachers we had, some of them seemed pretty burned out. And so I thought, let's just study biology. And I didn't really know what to expect from that. Like, what can you do with a biology degree? But the other thing that kind of got me into it is in the last couple of grades in high school in Germany, at the time at least, we had to do these huge presentations where you give sort of, I think they're supposed to be 20 minutes, but most people talk for about 45 minutes about a subject in a class that they chose. And then they wrote a whole paper about it. It was like 30 pages or something. And I chose HIV for biology. And I think that, that definitely changed it because I really like HIV as a virus, obviously not for the consequences for patients, but it's a very, very interesting virus. So I got very stuck on this, I'm going to work on HIV thing. So I did in my undergrad. We had to have a thesis at the end of so I went to the University of Heidelberg and we had to do a little thesis project at the end, and I did it in a HIV lab. And it was a pretty cool project. It didn't work out that well, but we had these chromobodies, which are fluorescently tagged single-chain antibodies that were supposed to target HIV capsid and sort of inhibit assembly. And so it was a really cool, very neat little interesting project. Unfortunately, like I said, it didn't really seem to inhibit assembly, at least not in my hands as an undergrad, right? And so then I went on to do my master's at the University of Heidelberg as well in molecular biosciences, but we had different majors, so I chose infectious diseases. And that was great. At the end of my bachelor, I didn't want to do anything else than get into that master's program because you would have a molecular biosciences kind of lecture where it's all kinds of subjects that are molecular biology. But then you'd have we had a virology part, we had a microbiology part, sort of bacteria, and I think parasitology as well. So it was all these things that, I was interested in. In my undergrad, I did biology and I learned, I had zoology, I had botany, I had all those things. But I really mostly just liked the viruses. And actually ended up taking essentially the same virology lecture 3 times in my second semester at university, where I couldn't actually take the class, so I just audited it, and then I took it again my 4th semester where I was allowed to take it, and then my master's had pretty much the same class again, because not everyone would have had a virology class at that point. But yeah, I like that. Yeah, I just liked all the viruses. So I worked again in my master's. We did rotations, and I worked in a lab studying foamy viruses, and in a lab studying HIV as well. That's where I did my thesis project on restriction factors of HIV. And during that time, we were allowed to do a rotation abroad, and I really wanted to do that. I really wanted to get a chance to get outside of Germany. And I saw a talk by my later on PhD advisor on Simliki forest virus and arboviruses in general, and I was just really hooked because in all these virology lectures, we never really talked about arboviruses. We talked about Togaviridae and alphaviruses, flaviviruses, but I feel like it never came across what arboviruses are and how important they really are. And I thought it was so. I went to Edinburgh and I spent 3 months there, and I loved it. I loved the lab, I loved the project, I loved the people. So I came back to do my PhD there as well.
With whom?
So I worked with John Fazackerly, who's a alpha virologist. During my rotation project there, I also worked with Alain Kohl, which you probably have met. And yeah, so I don't know, I was hooked. It was just, the city was great, the lab was nice, the science was great. So that's what I wanted to do. And then I got, I left the HIV world and I got sucked into arboviruses, and I have no regrets. So I worked on antiviral responses in tick cells. So that was kind of challenging because they're not really great to work with, those cells, for my PhD. And then I came to Colorado State University for my postdoc to work with Greg Ebel. And there I got all my mosquito experience, and I think ticks are really cool, but mosquitoes are so much easier to work with, and you can do large numbers and quick experiments,
So, and you've recently come here, only a few months, right?
Yeah, I think so. When I had some issues getting my visa in time and stuff, so I started here technically on August 12th, so about 6 weeks ago.
Nice. And I think you spent some time in Hamilton. Is that right? Or was it someone else on this?
No.
At the Rocky Mountain Labs?
Yeah, no, I didn't.
No? Did anyone else go to Rocky Mountain Labs? No? Must be another podcast. Because Marshall Bloom there does ticks.
Yeah, I know
cell
of them,
culture, but you don't do ticks anymore. So, okay. All right. So let's talk to you, Dave, since you're going to leave in about 20 minutes or so. You mentioned that your lab is interested in diagnostics.
Correct. Yeah.
So, I think you told us that you worked in a lab that did that. Is that where the interest came from?
Well, yeah. So I worked with I did a short postdoc with Tom Kozel, who's also in our department, and he's been working on pathogenic yeast for years and years. But then another project popped up that studied a bacteria called Burkholderia pseudomallii. And that one is a bacteria that basically has probably an 80% mortality rate if you're not on the right antibiotic. And if you are on the right antibiotic, in most locations, it's probably turns into about a 40% mortality rate. So this is a bacteria that's in the soil in Southeast Asia and South Asia.
Is this the necrotizing fasciitis?
No, it's not. But it eventually just leads to sepsis and death. And it's mostly a disease. The disease it's called is melioidosis, and very common in rice farmers, for example, because the bacteria is common in the soil, in rice fields, and anywhere it's kind of tropical and moist. And so the problem is it literally can take 5 to 7 days to diagnose the infection by blood culture.
And they would grow it out, right?
Grow it out.
Culture,
so take a sample of blood, put it in a blood culture bottle. Maybe 2 days later, you may get some growth, bacterial growth.
Is it aerobic or anaerobic?
It's aerobic.
Okay.
Yeah, and so you may get some bacterial growth eventually, but then you have to take a sample of that liquid and put it on an agar plate, pick a colony, wait another 24 hours, pick a colony, and then go through a lot of other steps to actually figure out what species of bacteria it is. So what the focus was at that point was, okay, let's try to figure out what this bacteria is shed or secreting during an infection, and then let's make some monoclonal antibodies that can target whatever is shed, so we could try to diagnose the infection more rapidly, maybe within a day or two instead of 7 days. So we actually, we started with some basic techniques. We identified a biomarker that was shed by the bacteria. We made a lot of antibodies. We did what's called an ELISA to capture the shed antigen within patient samples, blood, serum, things like that. But the key for our lab is that we started developing rapid diagnostic tests. So if you think of a home pregnancy test, we have all the equipment to actually manufacture prototypes similar to a home pregnancy test. And what a home pregnancy test does is basically just, detects a hormone that is produced during pregnancy, and it uses antibodies to do that. And so these are very common in infectious diseases as well, like for the rapid strep test, for example. So we built a prototype test for this disease, melioidosis, and we've been studying this for the last, probably about 10 years, and developing this technology, working with companies, and right now we're trying to get it through the FDA. So, hopefully a test that
this is a dipstick.
It's a dipstick test, yeah, basically.
And how early in infection can you get a positive?
That's a great question. I mean, a lot of the times, in these endemic locations in South Asia, a patient, after 10 days of not feeling very well, gets, winds up in the hospital. You can take a blood sample at that point and run this test. Unfortunately, there's not a lot of bacteria in the blood at that point. Well, at any point during a melioidosis infection, there's literally 1 bacteria per milliliter in the blood. So blood is not a great source. Other sources like a urine sample is a little better, actually. You may get, say, a 50% positive rate, on this for this dipstick test. There's other, skin abscesses can form as well. So just an infection on the skin that eventually can go systemic. A lot of times if you test kind of the fluid involved in a skin abscess, that has a lot more bacteria in it. So it depends on what type of infection, pulmonary, is it a skin infection, all sorts of things. But, basically it's a tool that the clinicians hopefully can use to diagnose the infection a little earlier. So that's our technology that we've used, and we probably now study about 10 different viruses and bacteria trying to develop rapid tests, everything from A to Z, really, anthrax to Zika. We're working on a project with Subash as well on Zika, trying to make some antibodies to Zika virus, but we have what we think is a really nice dipstick test for Ebola, for example. And with Ebola, obviously, you want to know as soon as possible when you're infected. So we developed a prototype test for that.
And these are antigen detection tests, right?
These are antigen detection tests. So a lot of dipstick tests will detect an antibody that the patient develops to a specific virus or bacteria. So they're looking, has a patient, producing antibodies to a specific bacteria or virus. Now that could say, they have been or are currently infected with this pathogen. We want to detect the antigen so we know that, this person is currently infected with this pathogen. So at that time. So we've got projects on Lyme disease, Ebola virus, a lot of other bacterial infections like plague and tularemia. And, basically the lab is kind of looking at patient samples, trying to figure out what microbial byproducts, bacteria or carbohydrates are shed during the infection. We want to know what those are. We want to then purify those, make a lot of it, make some antibodies, and then develop rapid tests for these pathogens.
So I guess part of the validation is you would take your test and compare it to whatever is done, culture, and see how many times you hit it when the culture hits it. Is that right?
Right, yeah. So usually with any diagnostic test, you're dealing with a gold standard. So for certain diseases like melioidosis, the gold standard is blood culture. So then you would say, okay, let's look at all those patient samples that were blood culture positive. How well did my test do? Right, and so for example, for our dipstick test for melioidosis, it doesn't do very well in blood because there's not a lot of bacteria in blood. We have to test other samples. So, but for Ebola, for example, we may compare it to a diagnostic test that is currently maybe FDA approved, or and look and see how does it compare with that one. What we want to try to make a test that's very sensitive, clinically sensitive. It has high. If this patient has Ebola, we want a high positive rate with this test, and we want to try to diagnose the infection a lot earlier because the current tests on the market, it's challenging to diagnose Ebola rapidly. A lot of times they get to the point where they're so sick they're about to die, then a test is positive. We're trying to make one that's a couple days earlier than that.
I've seen some of these filter pad based assays like you're talking about where the result is a QR code that you can then use a cell phone to transmit somewhere, so you can do it in the field.
Right.
Is that anything you're trying to do?
That's pretty common too.
Yeah.
So the technology is such that it moved from visually seeing a line or not, just like a home pregnancy test.
Yeah.
Now we're working with companies and other collaborators that they can snap in a lens or some sort of device onto a cell phone, and you can actually read the LFI right on the cell phone. Get the results. So a lot of times when you're looking at these tests, you're like, is that positive or is that negative? So let's let now the lens and the software analyze it and say yes or no. So there's no kind of user error.
Sure.
And after it gets a result, then it shoots it off to the cloud, notifies the proper authorities or clinicians and things like that to maybe rapidly respond to some sort of outbreak a little quicker. So that's exactly where the technology is going. And also it has the advantage of, instead of, for example, when you look at the positive or negative, a lot of times you're looking at an antibody that has a gold bead bound to it, and it's bound to the antigen. So all those tiny little gold beads line up on these dipstick tests, and that's how you see it positive or negative. If you can start incorporating like a fluorescent probe on the antibody, it may increase your sensitivity, say 10-fold, for example. So the reader then can, the device you're using can then detect the fluorescent signal, which is more sensitive than, say, the gold visual. So all sorts of exciting kind of point of care technologies that are developing.
So can you compare these antigen detection with nucleic acid detection? So we can do PCR. I was just at a European virology, clinical virology meeting, and they said in 5 years it's all going to be next generation sequencing.
Yeah.
So how do your assays fit in with that?
Yeah, good question. I think the nucleic acid amplification technologies are taking off. There's devices out there that are very powerful. Where we fit in, I think, is we can use our dipstick tests right at the point of care.
Yeah.
So, for example, you probably cannot have a $15,000 device that detects nucleic acid in every village in Africa. To head off an Ebola infection, right? But if you had these quick dipstick tests that would say, okay, we've got something going on here. Let's maybe, hey, now let's fly in some equipment and let's monitor the situation. Right. So,
or fly out some samples somewhere where there is a big piece of equipment.
Right. Yeah. And if you think about the recent kind of larger, the Ebola outbreaks that are currently going on, a lot of the samples are flown somewhere else. And so that tacks on another 2 or 3 days to diagnosis. So I think there will always be a a need for rapid diagnostic tests at the point of care in resource poor settings and in also every setting, really, right? But I think along with that, you're going to see better readers, to look at the test. You're going to see better connectivity, like you mentioned, to shoot this information to whoever needs it to get a rapid response. And there's also actually rapid dipstick tests that can actually detect nucleic acids as well. So you can do, say, like an isothermal amplification in the field, and then put that sample onto a lateral flow test or dipstick test, and you can detect the nucleic acid as well. So, potentially in under an hour, you can have a result.
Yeah, you may have seen these CRISPR-based assays, Hercules and so forth, that have come out last year, where you can do this in the field, apparently. Amplify and then use rapid detection methods, like you're saying. It's amazing, right?
It's really fun. It's a fun aspect of the job because I do like working on the translational products. Trying to get something from the bench to an FDA-approved product. We're close on one. We have a few others in the pipeline, but it definitely is motivating for me. Yeah.
So the influenza rapid test is notoriously inefficient, right? Can you do better? It's like 50% wrong. Okay. The rapid antigen, where you take a nasal wash and a dipstick in the doctor's office, 50% of the time it's wrong. Can you improve on? You should improve on it.
What would I improve on? I mean, I think you probably know better than me. But as far as like, you may see the inaccuracies due to the fact that a lot of influenza viruses are different.
Yeah.
And so one influenza test may not work for another influenza strain, for example. Is the amount of influenza in the sample, is there enough there to detect? For one thing, that's a problem we have with a lot of our assays, is there's really not enough bacteria or virus in a sample to detect.
Yeah.
I think that's a tricky thing. And yeah, I would say just trying to target more conserved regions on influenza. So you had maybe a pan influenza test that was very accurate. But at the same time, including some of these, nucleic acid amplification steps, I think, and building it into a rapid test may be helpful as well. So influenza is a field we have not gotten into because I know how competitive it is, and we kind of look at diseases that are neglected a lot of times. And that need some sort of better diagnostic. Yeah.
So in that sense, I was just at a meeting Monday of a consortium, and one of their viruses, they're trying to develop monoclonal therapeutics, a different thing, but Crimean Congo hemorrhagic fever virus is in need of better diagnostics.
I've heard this, yeah.
And I've heard that there, and also in Stockholm last month, I did a podcast with a guy who's working on that as well, and he said we need better rapid diagnostics.
And for antigen detection, or
yeah. I mean, anyone can make a PCR assay, right? But as you say, we don't have a machine everywhere. But if you could make a field, because there's a lot of infections in remote areas of Africa and Asia that could use it, right?
Right. And that's come up on our radar too. A lot of times we enjoy kind of working on those highly pathogenic, microbes. So that one has come up. Unfortunately, a lot of times it is what is the NIH, or more specifically NIAID, telling us to work on. And if the funding opportunity is not there, it's very difficult. It can be challenging to start projects on things that are not funded. But occasionally we do. We like to figure out what is the new virus that's coming, that's causing some outbreaks or infections. And if we can get samples and if we can make antibodies, then we try to start to do that early on to get a jump on it. So maybe when a grant opportunity comes out, we can say, hey, look, we've started this already, and hopefully, we can continue the work.
So at breakfast, most of the students this morning were from your lab.
Oh, okay.
So obviously you can attract students to work on these issues, translational issues.
Yeah,
as well,
I think it's. I really enjoy the lab. I enjoy the students. I think we get great students here, and these are. I think they're fun projects. I really do. I like. I think. I picture the students going home at Thanksgiving and talking about their little dipstick test that they made for Ebola or melioidosis, or we even use some of these antibodies actually as therapeutics as well. So we've taken some of these antibodies and shown in animal models that we can, save an animal from an infection, for example. And so maybe down the line we can convert some of these mouse antibodies into humanized antibodies and use them as potentially as a therapeutic as well. So that's kind of exciting too.
I know you have to go. I have one more question.
Sure.
If you hadn't been a scientist, what would you have done?
I probably would have. I think I would have really enjoyed either being a car mechanic or an architect, like a mid-century modern architect. I think.
Nice.
Yeah.
All right. Dave Aucoin, thank you so much for joining us.
Thanks for having me.
My pleasure.
This was great.
All right. You can just pop that off and go to your meeting.
Okay.
You're going to miss all the fun we're going to have.
I know. All right. Let me know how it goes.
Well, you can watch it. Thank you. So, Cyprian, last night at dinner, most of the students were from your lab.
Yes.
And so they're very excited to hear you.
Yeah.
On
Oh,
TWiV.
yeah. Well, a lot of them listen to TWiV and some of the other podcasts that you do, and so.
So, which herpes virus are you working on? Because I look at your website and your papers, and you've got both CMV and Kaposi's sarcoma, both.
We do both, and that's actually one of the things that I love, and I think it's one of our strengths is to actually work on. CMV is a beta herpes virus. KSHV is a gamma herpes virus. We also throw in some herpes simplex sometimes as a control, and so that's an alpha herpes virus. HSV is a whole nother story. So we don't work on that, but we do grow it and kind of compare.
It's wonderful when a virus is a control, right?
Yeah, I know.
That's what we've done with polio to a certain extent. It's our control.
Well, HSV it's so funny because you can get virus out in a day versus with
virus out of cells in culture, for example?
Yeah. So it takes about a day for HSV to replicate versus CMV. CMV, you infect it and come back 4 or 5 days later and you might get some virus out. But going back to what virus we actually work on, so we do work on CMV and KSHV, and really I like working on both viruses in a way to be able to compare and contrast. So what are some of the conserved processes that are found amongst both of those viruses, and hopefully we could extrapolate that to other herpes viruses, but then also how are they different? And they are very different viruses.
So CMV, most people are seropositive for CMV.
Yes, most people are seropositive.
What about KSHV?
So here in the United States and actually most European countries, it's about 10 to 15%.
Very low.
Yes, and there are some socioeconomic groups that are higher.
Okay.
But if you look at Africa and actually sub-Saharan Africa, it's 80%, 85% are KSHV positive. So definitely a very distinct geographic distribution for KSHV, where most other herpes viruses have a ubiquitous geographic distribution. And if we think of things like VZV, EBV, 100% of the population is positive.
So what is it about comparing the two that is of interest? Like what aspects of comparison are informative?
So two main focuses. One is on viral long non-coding RNAs. This is something that was initially for me a project as a graduate student, and it was one of those, I had two main projects, and then this was like the third project that I kind of got to work on when the PI wasn't necessarily paying attention to what I was doing. But for me, in KSHV, one of the things that I was always super interested in is this long non-coding RNA.
There's just one, or there are many?
There's a few, but this is the predominant one, and it's called PAN, polyadenylated nuclear transcript. It is the most abundant transcript during viral replication. It accounts for over, I think it's something like 70 or 75% of all transcript from the virus during lytic replication. And for a long time, nobody knew what it was doing. So, I mean, for me, it came back to the question of, for the virus, especially for herpes viruses, there's controlled regulation in gene expression. You have temporal regulation. And most of the time, these things aren't just being expressed just because. There has to be a function behind it, or, there wouldn't have been that genetic selection pressure to keep it.
It's not in latently infected cells?
It's difficult to say. Sometimes we pick it up, but it's the latency models are a little bit complicated because sometimes you have a little bit of lytic replication in the background. So we think that it might be expressed during latency, but again, it's hard to tease out what's going on.
So tell us, I guess you use cell culture models to model latency of KSHV, right?
Yeah.
So we know for simplex, the lat is the only thing made, nothing else. What's the situation for KSHV?
So for KSHV during latent replication, mainly there's, like Subash mentioned, there's LANA, latency-associated nuclear antigen. LANA is the protein responsible for tethering the viral episome to the host chromosome. It's also responsible for replicating the DNA during latency as well, along with host factors. So during latent replication, there's going to be a lot more host factors involved. So for pan-RNA, we started out, trying to look at its function, and it's really interesting because its ability to be so abundant. A lot of that work was done in Joan Steit's lab. Nick Conrad did a lot of that work, and so what they found is there's actually this nuclear retention element in the RNA. So it is polyadenylated, but it remains in the nucleus.
And it's not spliced, right?
Nope.
And is non-coding.
Nope, it's non-coding. There have been some studies looking at whether or not it could code for some small peptides, but those so far have been inconclusive. The other thing that Joan Steit's lab did is they actually crystallized the 3' region, and this was the first natural triple helix to be crystallized. So what happens is the poly A tail actually binds up into this helix at the 3' end, and so the poly A is sequestered in there, and that's going to help retain it in the nucleus and also account for some of the stability.
What's the promoter for this RNA? Is it Pol2 or?
Yes, it is a Pol2.
Can you knock it out, and is the virus able to multiply?
So that was one of our questions, was if you remove it, is the virus. So our lab, we actually made a mutant where we deleted, there's an overlapping reading frame, so we couldn't delete the whole thing, but we did delete the majority of it. And we saw that there was no subsequent virus production. So the virus is basically dead. Joan Steit's lab has also gone and depleted it using oligos. Same thing. So there is a definite defect in
lytic replication, right? Okay.
Without it. And so that's still something that we're still trying to figure out what its function is. One of our main hypotheses now is that it's involved in gene expression. And so we've found that pan-RNA is binding to chromatin modifying complexes and can modulate viral gene expression. And so this is really something that we continue to focus on. So we look at PAN's interaction with histones, histone modifying complexes, and then the viral protein partners, because some of the targeting of PAN to the viral genome is through other viral protein intermediates.
So you think it might direct proteins to the viral genome that desilence it?
Yes.
Right?
Yeah. To either directly, work on chromatin, histone methylations and things like that. And then also to help kind of set up the architecture in the nucleus that's going to help to promote that concentrations in the replication compartments that are going to be involved in gene expression. And so for when we look at long non-coding RNAs, then we also like to look at the non-coding RNAs for human cytomegalovirus as well. And again, the 4 most abundant transcripts for HCMV during lytic replication are non-coding RNAs. And so we're kind of looking at those to see what their function is. And we're coming at it in a very similar way. We knock it out, we see how the virus replicates. We also are overexpressing them and see if they have a function on their own.
So I guess part of this would be to ask what's binding these RNAs, right? What proteins and maybe other nucleic acids too. So is that something you do?
And that's actually one of the things that we did with PAN initially, is that we in vitro transcribed PAN. We put it onto a column. We then put over infected nuclear cell extract and did proteomics analysis to see what was binding. And actually that is what led us to, so one of the main binders was histones. The other one was a protein called ORF59. ORF59 is a viral encoded factor. It's the processivity factor. And so that's kind of that, so that was actually funny because it was one of my other projects as a graduate student, is to look at DNA replication. I was looking at the core complex. So for herpes viruses, they encode the core complex required to replicate the genome. And so it just happened that PAN was also interacting with the processivity factor. And so this is, again, something that my lab continues to work on, is the interaction between PAN RNA, the processivity factor, and how does that actually facilitate gene expression. And so we think that the processivity factor actually has a role beyond just DNA replication.
So do you understand why it's retained? You said there were some retention factors for the nuclear retention, is that right?
Yeah, so PAN is retained in the nucleus. One is that, so it's not spliced, but it is polyadenylated, and the poly-A, since it makes the triple helix, it doesn't seem to be interacting with any of the export machinery that would normally get it out.
Yeah.
Now, that's not true for the other, so for HCMV with its 4 non-coding RNAs, some of those do actually get out into the cytoplasm. And so actually one of the best characterized non-coding RNAs for CMV is one called beta 2.7. And this is something that John Sinclair's group and Matt Reeves, they actually showed that it targets to the mitochondria and prevents apoptosis. And again, there doesn't seem to be a homologue for KSHV in that respect. So these non-coding RNAs, well, we try to, again, kind of getting back to being able to compare and contrast how these viruses have evolved different non-coding RNAs is one of the things we're interested in.
So I guess one of the main drivers of export from the nucleus is being spliced. So if this is not spliced, that would automatically, because splicing marks RNAs for export, right? But if this is not spliced.
But herpes viruses do usually have their own kind of splicing factor that's going to help to export intronless RNAs out into the cytoplasm.
As most viruses do that export unspliced, aren't, like HIV and other retroviruses that need to export the whole genome without being spliced. And so those, so you have to suppress that in some way as well, right? So does this KSHV have such export? Does KSHV splice?
So some
mRNAs?
transcripts are spliced, but it does have a factor called MTA that promotes the export of intronless mRNA.
Got it. So last night I went to dinner with Veronica. Is she in your lab?
Oh, Vanessa.
Vanessa. Sorry, Vanessa. Shannon,
yeah.
she's in your lab. Salome.
Yep, Salome.
Ryan. Now he said he was a chemist, but he does something with
Ryan actually. So Ryan was one of our microbiology undergraduate majors, and as an undergrad, he started working in a chemistry lab, and so that was when he decided to go to grad school. He decided to keep with the chemistry, but they work on antimicrobial compounds. So.
And Jacob?
Yes.
Is an undergrad.
Jacob is an undergrad in the lab. Yeah.
And Jenna?
Oh, Jana, yes.
Jana, I get the name.
Yeah, it's okay.
Lucky I remembered this much.
Yes.
So Salome showed me on her phone, she has a picture of CMV
Yes.
infected cells where the DNA is labeled.
Yes, yeah.
And she said in non-dividing cells, you can label it because the label goes into the viral genome. Is that right?
Yeah, so what we do for CMV, and this is because we use primary cells. So for CMV, we can actually, they're primary fibroblasts, and we grow them to where they're confluent, and then we give them a couple more days. And so you have basically no cellular DNA replication going on. We then put on virus, and for CMV, DNA replication begins about 36 hours post-infection. At that point, she goes and we'll pulse label with nucleoside analogs. We use EDU, and from there you can use click chemistry to attach an Alexaflor to that EDU.
So you can see it.
So yeah.
Nice.
Yeah,
Very
yeah,
nice.
yeah. And so that's what her work has been on primarily. And then this gets back, we use HSV as our control there, because HSV, you can get DNA replication in about 6 hours.
Okay. You probably know that recently Pat Moore found lots of circular RNAs in KSV,
yeah.
KSHV.
Yeah.
I mean, hundreds and hundreds of them.
Yeah.
Are you interested in those? They seem fascinating, right?
Yeah, it's really interesting. I think, I had thought about whether or not, because they're formed through some back splicing events, and so I had thought about whether or not we want to just try to mutate those sites and then,
Yeah, there are lots of them apparently, yeah.
Yeah, that's the difficult thing, is that there's a lot of them.
And also we're learning that methylation, amethylation is, for some of them, can actually get them to be translated, because without a 5' end, you wouldn't be able to translate.
Yeah.
But it's all really amazing.
Yeah.
Just keep looking and you find more. Yeah. So you collaborate with Subash. Are you currently collaborating on something?
We do work on, since we work on same virus, like a sarcoma, so certain aspect we do work together, like WRF59. So we have shown that WRF59, this is a processivity factor. This can alter the chromatin, especially the arginine methylation on viral genome. So in latency, there is certain kind of methylation, and when reactivation happens, this WRF59 recruits this complex which can demethylate and can cause open chromatin. So that one area we have worked upon. And also WRF59, since this is abundantly expressed protein, it's quite high. I mean, we believe that this protein is there early during infection as well, although we see RTA, another immediate early protein, but at the same time, or maybe earlier than RTA, we see this WRF59, so we are trying to figure out what may be happening with this viral protein, whether it's chromatin modification, or like she said about pan-RNA, maybe pan-RNA or WRF59 binds and alter the chromatin structure early during infection as well. Maybe it silences the incoming viral DNA when it goes to the cells, and it'll establish this latency quickly.
Yeah.
So those aspects we are working on.
So you published a paper this year on this DNase activity of the SOX protein. You want to talk a little bit about that?
So the SOX protein, we know that this protein regulates mRNA level, but that work is done by Britt Glausinger at UC Berkeley.
Right.
But she did not look at the DNase activity, how DNase is important in terms of packaging the viral genome into the capsid. So we thought that DNase might be required for cutting these unit length DNA to package into the virion.
Because when it replicates, it makes
a concatemer.
Does it do it by rolling circle?
So later time it does rolling circle, but in the beginning, that's another question we are trying to figure out, whether it starts as a rolling circle or it starts as a theta model where there is a bubble, and when there is sufficient length or bubble is big enough, then it starts to replicate as a rolling circle. So we use this technique called single molecule analysis of replicated DNA, and this is like a combing of individual DNA molecule on a glass slide. So we can monitor the location of replication forks on individual DNA molecules. And looking at that tells us, if we assemble a molecule, it can tell us how the origin initiated and how that progressed. So in theta model or delta model, or theta model, not delta, theta model, it will be like going to both directions, but in rolling circle, fork moves in one direction.
Right.
So we are trying to figure out that as well, but it's a little complicated in terms of like when the circle completes, next round has to come in, and the lagging and leading strands, sometimes it causes overlap. So we are trying to figure out that, like how we can resolve that issue where we don't see the overlap, but we look at the replication fork only like with a single color, and that will not have problem in concluding whether the replication fork is moving in bidirectional manner or unidirectional manner. And that's an interesting question, and I think the electron micrograph has shown that it does start like a theta model, right? It starts as a bubble.
Like SV40, right?
SV40, yeah.
So for SV40, eventually it's resolved by topoisomerase.
Yes.
Is there some role in KSHV as well? Do you know?
Yeah, so topoisomerase is required. Some other group like Yannivan lab has shown that topoisomerase 1, I think, and 2 as well, they are part of the replication complex. So this protein, like immediate early protein RTA, this is the major factor in initiating the replication. This assembles the replication complex. And people have shown that there are multiple protein which binds to this RTA protein and probably helps in initiating replication as well as progressing the replication fork.
I see.
So maybe topoisomerase does that function when the fork is big enough, it chops it up, and then rolling circle starts at the time, but again, it's not proven, just the model.
And so when you're in the rolling circle mode, you're making concatemers.
Right.
And you're thinking that this DNA of SOX is cutting that?
Right. So that's what we showed, that if we mutate that DNA's activity, so the mutant was, the site was identified by Bert Glausinger, that if you mutate single residues, DNA's activity is gone. So we did that in KSHB genome itself, like we mutated the single amino acids, and there is no DNA's activity. And we asked the question whether we are generating a packaged virion particle where the virion produced are having the KSHB genome or not. So we found that, in fact, once you mutate that, there is a reduction, or not completely, I think it's totally, the number of virion produced which are infectious is totally down. So it does show that there is a problem with packaging, but we still don't know whether there are other protein like ORF69 or 67, those are other packaging factor, whether they are affecting the process or not. But we do know that mutating the DNAs or removing the DNAs leads to abortion or abrogation of virion production.
And you get very long DNA probably, right?
I do get long DNA. So we use this technique, again, like I said, single molecule analysis of replicated DNA. And we see that there is not unit length cut. There is a random, like longer than unit length DNA in there. So that's a suggestive that DNA's activity is working to chop it up into unit pieces.
Got it. So do you think the DNA is threaded into the capsid and then cut? Like for herpes, right?
Right. That's what we believe, that the other complexes which comes in, and the DNA I guess this goes in there and chops when they get packaged, when they're unit length. Some other machineries are involved. Like it's not as simple as we are thinking. Like maybe some other viral protein, they recognize the terminal repeat area where they chop it up and DNA goes inside there. So. That's what I think is similar, like maybe simplex, but we need to have a better experimental.
So one of your main interests is DNA replication and these kinds of questions, the resolution, the packaging, and so forth. And you mainly work on KSHV?
Yes, we do work mainly on KSHP and like litigraph DNA replication as well as latency as well. So latency is another area where virus replicates when, because in KSHP infected cells, those are tumorigenic cells, they divide. So when the cell divides, virus also need to divide to go on to the next divided cells.
Right.
So the mechanism wasn't known, like how the virus is replicating, and does it replicate more than the normal cells or not? So we have shown that the virus replicates using cellular machinery at the same time when the cell is dividing. And there is a close, I mean, some people, they call it like hostogen or something, that they're in close proximity of the host genome. It uses similar kind of machinery, or same machinery, and divides at the same time. But this protein, which tethers the viral genome, this takes those the divided molecules, or KSHB genome, to the daughter nuclei, to the mitotic apparatus is involved there. That's how the viral genome is pulled to the divided cells. So it's pretty interesting area for basic science purposes, like how this thing happens, like how Lana is working, whether it's working with another protein or it's doing majority of the work is done by Lana. So those are, I think, fascinating area.
For sure. But of course, herpes doesn't have this problem because neurons don't divide. So.
Exactly. Yeah. The gamma herpes viruses they have because they're tumorigenic virus. The virus induces the cell growth. So when the cell is dividing, virus need to pass on. And we know that the viral genome copies they don't get lost over time. So there has to be some or intricate mechanism where the virus is passed to the daughter cells in a very specialized or very programmed or very special manner that each cells receive the same copies when they're divided.
Does the viral genome encode its own DNA polymerase?
During lytic replication, yes, but during latency, it doesn't. It uses cellular replication or polymerases.
Because the genome is mostly quiet,
silent,
right?
Yes. Yeah.
I see. It's very interesting. So in latency, these copies of the viral genome are made entirely by host enzymes.
Yes, right? But the thing is, if we delete this protein, the latent protein, LANA, then you don't get replication. So it's dependent on LANA. Maybe it just tethers it or assembles everything.
Is it like a T antigen?
It's like a T antigen, right.
So it brings the replication
complexes together, yes, yeah.
Because SV40 only has T antigen, nothing else, and that's all it has to bring the cellular complexes, right?
Yeah.
And if the cell is not dividing, there won't be any genome replication of the virus, right?
Right. I think, it doesn't replicate at the time, yes.
So does the virus in some way make the cell divide?
It does induce proliferation, especially this LANA protein. This is an oncogenic protein, so it induces G1 estrangesin.
So that then you have the proteins needed to...
Yeah.
So it's very much like T antigen, right?
Yes, exactly.
So do you see, when you get tumors, is LANA constitutively produced?
Yes, LANA is the one which is there in all the tumor cells.
And if you just put LANA alone, does that cause a tumor?
No, it doesn't cause a tumor, but it can induce like various cellular pathways, which are like indicative of tumorigenic, tumorigenicity. But it cannot transform. The virus itself cannot transform. When KSHB, entire virus, not just the protein, LANA protein, KSHB doesn't transform the cells. Although GPCR has been shown that, like another protein of the virus, that is shown that it can induce tumorigenesis, but the transformation is one of the things like KSHB cannot. But recent paper came out in PNAS from the guy in Wisconsin, not Don Geenum, this
was it Bill Sugden?
Bill Sugden's lab. He showed that if you have EBV in combination with KSHB, it can transform the cells. Like there is a different epigenetic marks and changes happens in the cells where if both the viruses are there, cell can be transformed. But KSHB by itself cannot. But EBV, like the other homologue of KSHB, it can transform the cells if you block the T cell function.
So in cell culture, KSV cannot transform cells,
No,
right?
it cannot.
But in people, it causes tumors, right?
Right.
And so you're suggesting maybe EBV is part of that or something else, yeah? Okay.
Yeah, that's what the conclusion of the paper
yeah.
was that, like,
Yeah.
everybody, not everybody, but almost 95% of the people, they are positive for EBV.
Yeah.
And if they get KSHB,
Okay.
both the viruses probably enhance or they can induce tumorigenesis and the cells
Okay.
are transformed at
Cool.
that point.
Very
Yeah.
cool. Let's talk about mosquitoes.
Okay.
So, and you're interested in defense mechanisms, innate defense
Yes,
mechanisms of mosquitoes. So first, are there mosquitoes in Reno?
There are mosquitoes.
There are, for sure.
There's actually
Not just in your lab.
no, no. There are actually mosquitoes in Reno, and I guess what Dave was referring to is that you don't have these nuisance mosquitoes in giant numbers as you would in Minnesota or something. But
New York, New Jersey, yeah.
yeah. But
What kind of mosquitoes are they here?
So I don't know the whole mosquito population, but I know that there's for sure Culex mosquitoes, which are the vectors of West Nile virus in the US. And the reason why I know that there's Culex mosquitoes is because there are cases of West Nile in this area.
Okay.
So I know that so I obviously just recently moved here and haven't figured everything out, but I know that the city of Reno has a control program where they catch mosquitoes and they're screened for West Nile. So there must be mosquitoes. They're probably more around the agricultural areas. There's probably other mosquitoes around the Truckee, where it's a bit wetter. Yeah.
Are there Aedes species?
I mean, definitely not Aedes aegypti or Aedes albopictus, but there might be
Others, okay.
kinds. Aedes vexans is a common sort of nuisance mosquitoes around
All right.
parks and stuff.
You have mosquitoes in your lab, though, right?
I don't yet.
You will.
It's a
plan.
I will. They're currently planning the insectary, and the plans have been made. So hopefully, in a few weeks, they'll start construction.
And what kinds of mosquitoes will you breed?
I will focus on Culex, but there will also be projects where I work on Aedes aegypti or Aedes albopictus.
Okay.
But based on Culex being sort of the main vector of arboviruses in the U.S., really, apart from southern Florida and southern Texas.
Right. Where you would find Aedes aegypti.
Yes, exactly. Where you worry about dengue and other tropical diseases, but what most people probably need to worry about is things like West Nile, or now Eastern equine encephalitis virus is spreading, has a lot of cases in the Northeast, yeah. So all these viruses can be transmitted by Culex, and I consider Culex to be a bit understudied, mostly because just the reagents have been generated more for Aedes and Anopheles as such, and Aedes admittedly are a little easier to work with in some aspects because they have different life cycles, so you can maintain them easier. And a lot of the focus has just been on Aedes aegypti and Anopheles mosquitoes. So it's harder to start getting reagents and materials and expertise in something if no one starts.
Sure, of course. So aside from West Nile here in Nevada, would there be WEE? Or has that ever been seen?
Yeah, that's not really seen in the field anymore.
Anything else here?
Here you would have, there's occasional cases of St. Louis encephalitis virus, which is, I guess, a similar clinical phenotype to West Nile. And California encephalitis virus, which is a orthobunya virus. They're rarer, but I looked at the history of viruses in Reno, and they do occur occasionally in this area, not necessarily Reno, but maybe surrounding parts.
Okay.
And I'm sure that some of these might be becoming more, what's the word? Frequent, I guess.
Sure. So you have published on small RNA responses, culisines.
Yeah.
So give us an overview of what happens when a mosquito's infected.
So
takes a blood meal,
this is why I think arboviruses are really cool, because you have to think about them as being confronted with two completely different host systems, right? Like we have humans or birds or any kind of vertebrate that is their vertebrate host, but then you have the arthropod vector, and they don't just get transmitted mechanically, they really have to make it through the whole vector. So essentially, when a mosquito takes a blood meal on for example, West Nile, most of the time it's an infected bird. They take a blood meal, and the virus enters the midgut of the mosquito, and now it has to enter the midgut epithelium, the sort of a single cell layer that surrounds the blood, makes up the midgut. And the viruses have to make it from that point, from being in the midgut, to the salivary glands and into the saliva. So otherwise, they just infect the mosquito, and that's it. So what happens is they infect the midgut epithelium.
And there are some viruses that just infect mosquitoes, right? And that don't
there are viruses that just infect mosquitoes. They obviously wouldn't get into the mosquito through a blood meal. They would get into the mosquito through vertical transmission or other
plant sap.
Plant sap, exactly, other mechanisms. Okay. Yeah. And luckily, those are viruses we don't have to worry about as much. But the arboviruses have to then get out of the midgut again, replicate in the body of the mosquito, sort of in a disseminated infection, infect the salivary glands, and then be produced in the salivary glands to high enough titers that you can initiate an infection in a host. And there's a few mechanical barriers to this. So some viruses may not even make it into the midgut epithelium, just based on receptor availability or other factors.
Could they be digested in the gut?
Yeah, they would probably just be digested. And
what is the gut of the? What is it called?
The midgut? Or,
midgut.
Yeah.
Is it low pH, high pH, lots of enzymes?
Yeah, there's a lot going on, a lot of, I don't...
Do you know
actually,
what the pH
I actually know about pH, I have to think about that.
Well, it must not be acidic, otherwise these viruses would uncoat.
No,
right?
yeah, but there is a lot going on, and there's a lot of microbiome that can play a role in, in this whole process.
Fungi too, we've talked about that on TWiV, they can help.
And they digest their meal for, about 3 days or so. So that I think, and they form kind of a, let's call it like a layer around the blood meal. So if that happens, the virus probably wouldn't get into the cells either. So essentially they have to get into the midgut cells very quickly. And what some older papers have shown for example, one of the encephalitis viruses, that what we sort of assume is that a few cells get infected initially, like just a couple in the posterior end probably of the midgut, and then they initiate the systemic infection. And there's so many differences too. This is also why I like arboviruses, because I can work with. It doesn't really. I'm not, you work on CMV and KSHV, but I can work on 3, 4, 5 different groups of viruses, looking at similar processes, but they are very different, and alpha virus replicates really quickly. So chikungunya virus in the mosquito gets, from the blood meal to the saliva in 2 days, essentially. Whereas, or not always, but it can. And with viruses such as West Nile, it takes, 7 days until you see some mosquitoes that have a virus-positive saliva.
But mosquitoes don't live very long, right?
Yeah, they don't.
14 days, more or less, or longer?
No, they can live sort of 4 weeks. I mean, it depends on the field, on their conditions and stuff. But I think, so if we keep them in the lab, they can definitely survive 4 to 5 weeks. And I think that a lot of that depends in the field on like temperature and predators and all those.
Yeah, they can be eaten, right?
Yeah.
So only female mosquitoes take blood meals.
Only female mosquitoes will take the blood meal.
And they have to take more than one in their lifetime to transmit a virus, right? Okay.
Yes, and
so
that
do we know how many they take in a lifetime, blood meals?
That depends again on the species a little bit, and whether they can take a full blood meal or they get interrupted. So, for example, Culex will often take a full blood meal, and you can see that they're really engorged and have a really fat abdomen, essentially. And then they will, process the blood, and the blood is really taken up to lay eggs. That's the main reason why they take blood meals. There's also some Culex species where you have a lot of egg lays without taking a blood meal. One of my colleagues at CSU, she took mosquitoes from the field, Culex tarsalis, and a lot them they were laying eggs, without having taken a blood meal.
They just drink plant sap, and that's enough.
Yeah. And Aedes aegypti, for example, is kind of more known to take a lot of small blood meals. So, which also means that they're maybe a better vector for a lot of things, because they will take a small blood meal and then might not get enough nutrients to lay eggs, and then 2 or 3 days later take another blood meal. So at that point, you get all these smaller blood meals that are all opportunities to transmit virus in a way.
Okay. Apparently having a blood meal is a real shock to the mosquito, I read.
Yeah.
We did a paper where they studied these mosquitoes that don't take blood meal. They made them take blood meals, and you get a transcriptome of all these heat shock proteins because just think of it, there's a warm blood coming in and they have to deal with it, and it's amazing that so many do actually. Actually, there are probably more non-blood. Right?
Well, I don't know if they're more. Because in terms of species, the only, genus that we know that really doesn't take blood meals, that we know doesn't take blood meals, are Toxorynchides. So that's a different genus. Right. But so what I was referring to is a species of mosquitoes that does take blood meals, but occasionally doesn't. They're known to take blood meals in our vectors of West Nile. But still, you will find them to be able to lay eggs without a blood meal.
So when the virus is replicating in mosquitoes, what's the response?
Okay, so there's a lot going on. Essentially, you have to think about it in a way that we want to get rid of viruses. When we get infected, we live 80 years or so. So our main response is to get that out of us if we can. But mosquitoes, as you said, have a short lifespan. So what they want to do is just kind of control the virus, just keep it at a low so that it doesn't impact with their physiology for, a few weeks, and then they die anyway. So, So
at low levels, the virus is not going to hurt the mosquito.
No, and that's why we have all these insect-specific viruses too that are essentially not pathogenic to mosquitoes. They just kind of replicate and they tolerate them. But so what happens is when an RNA virus enters a mosquito cell, is that one of the first things an RNA virus is going to do is generate double-stranded RNA as part of its genome replication. And double-stranded RNA, as most people probably know, is foreign to a cell. It doesn't normally occur in the cytosol of a cell. So it's recognized by an enzyme called dicer2, that essentially chops up the double-stranded RNA into 21 nucleotide small RNAs, siRNAs. And these siRNAs are then used by the RNA-induced silencing complex RISC to target viral genomic RNA or antigenomic RNA. So you essentially have a mechanism that is sequence-specific, where you directly target the viral genome as an antiviral response. And it's very efficient, but it doesn't completely get rid of the virus. It just, like I said, kind of keeps it at a low. If you get rid of the response, the mosquitoes will essentially die. There's some. So
you can get rid of dicer or argon.
It's really bad for the mosquito. And then there's a lot of other things that I'm interested in studying, is the more complicated mechanisms of RNA interference that aren't just, dicer, argon out, and what we call exogenous siRNA response. There's also there are other RNAi responses, for example, the PWI interacting RNA silencing system, and so we call those piRNAs. And I think there's a lot of publications that came out over the last sort of 6-7 years on piRNAs in mosquitoes, but it's very unclear whether they really play an antiviral role and what impact they actually have on virus replication. So what people have found in Aedes aegypti is that if you infect, say, for example, with an alpha virus, because it looks very convincing with an alpha virus, is you get this, and you profile their small RNA responses. So you sequence everything that's between 19 and 32 nucleotides of RNAs in the cell. You'll find a giant peak at 21 nucleotides, and those are your exogenous siRNAs. And then you also find a peak that's around 27, 28, 26 nucleotides, which have a signature with always a uridine at position 1 and an adenine at position 10 in the negative strand and the positive strand correspondingly. And those are piRNAs. So they are generated through a mechanism independent of dicer. And normally piRNAs are part of our mechanisms to control retrotransposons in the germline, essentially. So you get, in the germline, if you have retrotransposons being expressed and integrating themselves all over, then you're just destroying your genetic information. So we get piRNAs that target these retrotransposons and kind of keep that under control. But in most animals, really, they're only expressed in the germline and not in somatic cells. But in mosquitoes, there's this huge expansion from, 2 to 3 PWI genes to like 7 that are also partially expressed in somatic tissue, so in the midgut of a mosquito, for example. So I still think that they have an antiviral role, but the fact that these are being generated, hopefully I'm not doing anyone wrong, but as far as I'm aware, no one has really shown that when you get rid of that system, when you knock them out, you knock them down or so, you get any impact on viral replication, or very little. So they're being generated, but we don't really know if that is important. I mean, you would assume so if you have this expression in somatic cells, that they're doing something important. But
so that's one of the things you're interested in.
It's one of the things I'm interested specifically in, again, in Culex mosquitoes, because everyone's kind of looking in Aedes. But
and which viruses would you use?
So for West Nile, California encephalitis virus as sort of a Bunia virus, and then probably some Synbis. So I'm trying to get an idea of the different groups, trying to compare and contrast and see if it's maybe a mechanism that has evolved against a specific group of viruses and then is just a byproduct for others. And I haven't, in the paper that you mentioned, we haven't actually found any evidence for these piRNAs against West Nile virus. So they might not be generated to flaviviruses.
So is your lab set up and you're doing experiments yet?
No.
Not yet.
No, it's been kind of slow getting everything to the point. I think later today I might be able to order equipment and stuff like that.
Good. Great. Cool.
We'll see. I do have an undergrad who's going to start working with me, and I'm recruiting a technician who's going to start working in October as well.
And of course, you're working in the lab as well.
Yeah, eventually, when there's things
to do. Yeah.
Yeah, I have pipettes. So.
Subash, do you still work in the lab?
A little bit.
A little bit?
Not a lot. Not like Cyprian does.
So Cyprian, you work a lot in the lab, right?
I love being in the lab.
You think you always will, forever?
I think it's what I love to do, and it's what I look forward to every single day. I mean, it's still every day I love to go and look at the cells. I love to see infected cells. We have viruses that express GFP, and they are also tagged with DS red on certain proteins. And I love to go.
They're beautiful, right?
They're beautiful. I love to see the spread.
Great.
It's just,
You also teach, right?
Yes, yeah. So I actually, in the spring, I teach the undergrad virology course. So I have a lot to thank for you, because we use your textbook, and then I watch your YouTube videos, and it definitely helps me to organize the information and be able to present it to the students. So I thank you for putting all of that out.
Do you teach any other courses?
I do some guest lectures here and there.
But that's your main virology. How about you, Subash? Do you teach?
So I taught some virology lecture to med students.
Medical students.
Yeah,
right? Yeah.
But things are changing a little bit here in terms of teaching by instructors. So we'll see how it goes.
Yeah, I used to do a lot of medical student teaching when I first went, but now it's all done by clinicians at Columbia. I teach an undergrad virology course, which I don't have to do at a medical, so I don't have to teach any.
Yeah.
We don't have undergrads where I am. I go to another campus, but it's important to teach them virology.
Yeah, no, it is nice.
Columbia did not have a virology course for its undergrads. Isn't that ridiculous?
Yeah.
So that's why.
So 5 years ago, Nevada didn't have a microbiology major in the whole state.
Yeah.
We were actually only one of 2 states that didn't have a micro undergrad major. And so that was something that our former chair, Greg Perry, started.
Good. Now, you're probably getting a break from teaching.
I have a 1-year break from teaching, but because the biochemistry department is kind of smaller and we have a big major, I'll have to teach every semester. Yeah, well, I'll teach molecular biology, which is good because I'm much better at molecular biology than biochemistry.
Actually, and maybe you'll do a guest lecture in virology in the spring?
I'd be happy to.
Okay.
I would love to teach virology, but sadly, because it's out of the major,
You can talk about this.
It'd be great if you could come in and give a lecture on arboviruses.
For sure.
So we have had on TWiV Raul Andino, who works on insect defenses, Carla Sally, Who else? I'm going to forget people and insult them. There are not too many people in this field,
No.
right?
Well, those are, the big ones for me.
Yeah. And Greg,
Actually-
your old PI, Greg Ebel does it as well, right?
Yeah.
We haven't had him on. One last question. If you hadn't been a scientist, what would you have done?
Oh, that's a hard question because I really always wanted to be a scientist. I think for a little while when I learned about radioactivity, I would have said nuclear physicist, but for the most part, I always wanted to be a scientist.
Subash, what about you?
Thinking back, I think I would have been in public service or something.
Okay.
Yeah, if I was not a scientist, going to serve in some public office.
What about you?
I think I wouldn't have strayed too far and probably been a veterinarian or something.
vet.
That was something
That's cool.
I was considering.
I asked Joan Stites that question. I forgot her answer, but it was really good. Something like a librarian or a bookseller, maybe. Or, I shouldn't say, I forgot, but you should go listen to her. We did a TWiV a couple of years ago at ASV with her.
Oh, okay.
And because Rich Condit had worked with her, so that was really fun. And RNA or DNA?
Ooh, that's another tough one. I love RNA.
RNA or DNA?
DNA.
RNA.
RNA. I always love the response. Okay, that's special TWiV at University of Nevada, Reno. You can find all our podcasts at microbe.tv/TWiV. Questions and comments, TWiV at microbe.tv. If you like what we do, consider supporting us financially. We do this all on our own. We get very little support except from our listeners. So go to microbe.tv/contribute, where we have mechanisms like PayPal and Patreon, where you could give us just $1 a month, and that would really help us a lot. My guests today at the University of Nevada in Reno, Dave Aucoin has left. Cyprian Rossetto, thank you so much.
Thank you.
Thank you for inviting me as well.
Thank you for coming here.
and arranging everything. Subash Varma, thank you so much.
Thank you. Thank you for coming here.
And Claudia Rooker, thank you.
Thank you so much.
And
for
good
having
luck.
me. Thanks.
We'll get you when you've been here for 5 years and talk about what you've done, right?
Sounds good.
That would be fun. I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.